The title of our presentation is Plastic, the New Superfood. The problem is that there are 51 trillion microscopic plastic particles in the ocean. This outweighs approximately 1,345 adult blue whales, and it outnumbers all of the stars in our galaxy. Microplastics are particles of plastic smaller than 5 millimeters in diameter. Polyethylene terephthalate is one type of microplastic. It is a petroleum-based plastic that is strong, durable, and easily processed. It is commonly used in the production of synthetic materials. 50 million tons of PET plastic are produced annually, and anywhere from 5 to 12 million tons are thought to enter into the ocean each year. Microplastics have profound impacts on animal health and can cause monumental problems to the marine environment. The small size of microplastics allows them to affect organisms as small as zooplankton. Biomagnification of microplastics, how it affects us. Microplastics are initially consumed by very small marine organisms like zooplankton. As predators consume zooplankton, the microplastics begin to accumulate in the stomachs of the animals where they can leach toxins. Animals cannot digest these plastics, which leads to biomagnification across the marine trophic levels, including top predators like humans. So this issue affects all organisms. What can be done? Recently, a new type of bacteria, Aedenonella sickensis, was found that can naturally break down PET plastic. We intend to harness this bioremediation to help solve the problem of oceanic microplastics. Aedenonella sickensis cannot live in saltwater environments, so we intend to transfer the two genes responsible for this breakdown into E. coli, which is one of the world's best studied bacterial species. This specific combination of plastic degrading genes has never been placed into E. coli before, so this is a new and innovative concept. Our solution. Aedenonella sickensis has two genes involved in the hydrolysis of PET. Gene ISF6-4831 codes for PETase, which is an enzyme that naturally breaks down PET into monoterephthalate. Gene ISF6-0224 codes for metase, which is an enzyme that breaks down MET into products used by the cell as a major source of carbon and energy. Our proposed solution is to insert ISF6-4831 and ISF6-0224 genes from Aedenonella sickensis into E. coli, which is our model organism that reproduces more quickly, is more durable, and can survive in seawater. Inserting these genes into the E. coli will allow it to break down the PET plastic that are in our oceans quickly and sustainably. Our solution is feasible because E. coli is susceptible to horizontal gene transfer, giving it the ability to accept the genes from Aedenonella sickensis.
Next, I'll be explaining the step-by-step process for inserting our two genes into E. coli. The first step is to use PCR to isolate the gene that produces PETase and the gene that produces metase from Aedenonella sickensis. Primers are needed for this process and are listed on the right. The vector we have chosen is PET22B. The next step is to cut the DNA with the restriction enzymes we have chosen. They are HindD3 and EcoRI for the PETase gene and XHO1 and NCO1 for the metase gene. They then will be annealed together by DNA ligase and the vector will be complete. The next step is to get the vectors into our E. coli cells. In our case, we are inserting two separate vectors due to the two separate genes needed to break down microplastics. We will insert our vectors via electroporation, which is essentially using a pulse of electricity to get the E. coli bacteria to take up the plasmid. We will use plates with an ampicillin marker to identify cells that contain both our plasmids. If they contain the plasmid, they will live. If they do not, they will die. We will then run a PCR and gel electrophoresis to determine if both of our vectors are present in the E. coli. Next, a Western blot will be done to determine if the proteins, PETase and metase, are being expressed in the E. coli cells. When the insertion and identification processes are complete, we will introduce our transformed E. coli into a laboratory-controlled environment containing PET plastic. The E. coli will be exposed to the environment for 48 hours, and the amount of PET present before the E. coli were introduced and after will be measured. The positive control in this test is Idenella, and the negative control is non transformed E. coli. If the amount of plastic present after the E. coli have been exposed for the allotted amount of time is less than when we started, our project was a success. If our transformation was successful and we introduce the E. coli into the ocean, we will make the bacteria dependent on a food source that is phosphorus and nitrogen based and not present in their environment that only we can supply to help us remain in control of the organism. Next, I will explain the vector diagram. So here's an image of our vector diagram on the right. As I said before, we chose the PET22B vector. The multiple cloning site is indicated with a bright blue font. Our restriction enzymes are found here and are indicated with blue boxes for the PET ACE gene and red boxes for the MET ACE gene. We decided to use a T7 promoter as it is successful in bacteria. We have the AMPR gene to identify transformative cells, and the origin of replication is also labeled. The chart on the left indicates the other items present and their functions.
Evolutionary context. So this is our phylogenetic tree, which shows bacteria Ideonella sakaiensis in comparison with some other bacterial species that are known to produce plastic-consuming proteins. So this is basically showing that E. coli is ideal for us because they're so close in comparison. Our main concerns for ethics are that our genetically modified E. coli could disrupt the ocean's ecosystem. The E. coli could spread outside of our target area and have nothing to control their growth. The E. coli could deplete the oxygen available to other marine organisms. The E. coli could harm the marine life by infecting them. The E. coli could degrade other plastics like plastic boats, buoys, or scuba gear. And we don't know the effects from inserting this gene into E. coli, so it could be detrimental to their health. And lastly, humans would be relying on some other organism to clean up our mess and not keeping it as our own problem. So to address these issues, we are going to provide a food source of a phosphorus and nitrogen based compound that we give the bacteria in order to make sure that they do not leave the region, otherwise they will die. The E. coli usually doesn't affect cold-blooded animals, so they most likely will not harm marine life. And lastly, E. coli will be most efficient in degrading microplastics, so human efforts are still necessary to clean up larger plastics. So in the future, we could determine a way to enhance the function of this gene so that it can break down fully intact plastics. More research towards enzymes that could break down other components of plastic, such as nylon, polypropylene, and polyethylene. And then we could also introduce similar approaches into landfills to target plastics before it reaches and starts affecting the oceans.